Okay, everybody, take your seats. We're going to start. As you can probably tell, I am not Carlos. I'm Lionel Christian. I also have a weird accent, slightly different from Carlos's. And before we jump into the topic of today's lecture, which is going to take us through many lectures and really through the journeys proteins take in the cells, I want to give you a few advices which I give students every year, every semester actually, because I also teach developmental biology. And that's about you guys succeeding in this course and in any other course. And it comes from the field of learning and memory, right? So how do we learn? How do we remember things? In other words, how can you adjust your own behavior to maximize your chance of success in this course and in any other course, really? So one of the first things is attention. So it's good for you guys to be here. Attendance helps because it helps you keep this pristine time to be in class and pay attention to the lecture. And what I mean by attention, it also means reducing all distractions. That means if you tend to go to a lecture but then have your Facebook, Instagram, and whatnot going, this is distracting. And it's basically a proven fact that it reduces your ability to engage with the material and eventually learn, right? So that's the first advice I would give is really pay attention, be at what you're doing. The second advice is to try to be active. Now this is harder because we are lecturing at you. So the temptation, and I've been there and I go to talks and sometimes it's easy to doze off. But if you're active, so if you're trying to, and that comes with attention, to engage with the material, to think about questions, to think about the implications of what we're telling you, somehow it helps learn it. Okay, so that's the second advice. And the third advice I'll give you is more about how memory works, how memories get consolidated, which also is an established scientific fact, is through repeated exposure to the material. So what do I mean by that? You come to lecture, you pay attention, you engage. Then you move on, you have another class, you go to lunch, you go to exercise and whatnot. You put it away. In the evening, you look back. And again, you look back being engaged, being at what you're doing. And then you put it away. And then again at the end of the week. And then again at the end of the next week. So by the time you study for the exam, you've seen this material with undiluted attention for the third or fourth time. And now it will really feel familiar. Okay, so these are the three main advice I like to give students to really help you succeed in the course, which is pay attention, be at what you're doing when you study or in class, try to engage with the material, and finally expose yourself to it. Learn it, pay attention to it multiple times. Let go, come back, let go, come back. And you will have a good command of it. All right, enough of this. Now we're going to dwell into this topic. Or maybe just one note. Since today I only have one lecture, I'm going to have office hours on Friday afternoon from 3:30 to 5. Usually I'm going to try to aim for Thursdays and Tuesdays, 3:30 to 5. And if changes are needed, I will let you know. And also, I'd like you guys to contact me in advance to set up some kind of appointment. Okay, so enough of housekeeping stuff. Let's dwell into the topic. So we're going to discuss a lot in these few lectures the trafficking of proteins. So basically cell compartments and how proteins, which are necessary for these compartments to work and to exist, are trafficked and transported to these places. So starting very broadly, you will hopefully recognize these eukaryotic cells in cartoon forms, which are marked by the presence of what? A nucleus, right? Eukaryotic, a big nucleus, which contains all the genomic DNA, whether in animal cells or in plant cells, right? And they have a lot in common. So they have, of course, in this nucleus we talked about, it's delimited by a nuclear envelope, okay? So two membranes, You already started seeing that. And then that nuclear envelope forms a continuum with another, series of membranes here, the endoplasmic reticulum. Very complex system, which is essentially two compartments, and we're going to focus a lot on the endoplasmic reticulum today, right? But of course, you know that there are more organelles, so to speak, in the cell, and for instance, here you can see very clearly, you know what this one is? The Golgi apparatus, that's right. So we're going to have one lecture at least focused a lot on the Golgi apparatus, how it's established, how proteins are dynamically transported through it. Other organelles like these guys or these ones here, chloroplasts and mitochondria. You've seen those with Carlos. They are where ATP is synthesized and where a lot of energy transfers happen. Okay, but today we're going to start to focus on this protein trafficking business. So I want to clearly highlight the first order of business that the cell has to achieve here, which is where do proteins come from? Where are proteins encoded? In the DNA. And the DNA is in the nucleus. Okay, so this is where the information is to start with. But of course, you've seen molecular cell 1. Everybody has taken molecular cell 1, right? Oh, actually, you got used to the French accent with Claude, maybe. Did you? Okay. Well, mine is, well, anyway, let's not do that. So you know that transcription happens in the nucleus, that there's a copy made of those genes, simplified somehow through splicing, and these mRNAs are exported into the cytosol. Because this is where translation happens, right? There is no way around it. Translation happens here, in the cytosol. And you've already seen, I hope, these big blobs here. What are those? The ribosomes, right? So they are made in the nucleolus. In the nucleus, they are exported as these complex machines and further assembled on the mRNA, on the mature RNAs, ready for translation. So we're not really going to talk about the details, the molecular details of translation here. We're really going to focus on what happens to these proteins that are being made. Because if you pause here for a second, one of the key problems that the cell has to solve, and I want you to think about, is how do you create all these compartments we talked about? How do you maintain them? How do you make them work with specific proteins? Whereas all the proteins initially are made in the same compartment in the cytosol, right? So it's essential that you have this trafficking, this addressing of specific proteins to specific places. And that's what's going to keep us busy for a few lectures, actually. So one mode is the most straightforward, if you will. You synthesize a protein. So synthesis happens first. Then the protein is present, fully formed, sometimes even well folded in the cytosol. And there is a so-called targeting sequence. If this is not a cytosolic protein, so a protein that's resident to the cytosol, can you think of some of those? Proteins that remain and basically work in the cytosol. Some you've seen with Carlos. Yes?
I'd say like enzymes using glycolysis.
Absolutely, that's right. Glycolysis happens in the cytosol, so the key enzymes, like for example, do you have an example? Phosphofructokinase, that was the one I was thinking about. Do you read my mind? No, good. So unless there are such proteins, such cytosolic proteins, they have to be shipped to a particular location, be it the chloroplast, the mitochondria, the peroxisome. So we're going to talk about how this is done. And we may not talk, so in the syllabus, it's scheduled to be next week already. Actually, we'll rearrange to focus the first part on the secretory pathway. So we're going to talk about those a bit later. But can you think about a few different proteins that need to be targeted to the mitochondria? Now, this is a tricky one. So let me just say that at this moment. You remember the mitochondrion, it's special, right? What's special about the mitochondrion? I mean, there are many things, but one of them. It has its own DNA, that's right. So that means you could say, well, for the mitochondrion, this does not really apply because it can synthesize its own protein from its own DNA and RNA and so on. And that is true for a few of these proteins. But you may have heard of the concept of endosymbiosis, whereby mitochondria a very long time ago became part of the eukaryotic cell. And part of this meant transferring some of the genes to the nuclear genome. So now the eukaryotic cell and the mitochondria are completely codependent in terms of energy, in terms of composition. Okay, that's not even what I want to talk about to you today. Okay, the second part, and there's a key difference. I mean, there are many key differences between what happens here and what happens there. And one of the concepts I will emphasize a lot today, and I really need you to grasp and understand and remember, is the concept of co-translational translocation. What does that mean? What I mean by that is that here, like I told you, the protein is synthesized first, and then it is shipped to the correct location. Right? That's true for all these organelles. But in the ER and en route, I don't know why I say French word with an English accent, en route, to the Golgi and the secretory pathways to the plasma membrane. So proteins have to be translocated first in the ER, and this happens co-translationally. That means they are being passed on into the ER as they are being synthesized. Okay? The two processes are coupled, translation and translocation. So translocation means the proteins moves across the ER membrane. Okay, and that's what we're going to really focus on today. And then as we'll see in a few more lectures, the proteins then has to be trafficked, undergoes a bunch of modifications, and depending on the exact signal that it carries, it will be shipped to either remain in the reticulum or move forward to the Golgi and so on. Okay, so in broad terms, you can break down the problem of, okay, how is a protein trafficked, shipped to a particular place into these simpler itemized questions, if you will. One of them is, what's the nature of the signal? So in other words, that's usually in the protein, as we'll see, and what is it? You know, what's the zip code in the protein? Because it has to be encoded in amino acid sequence. The second is, what's the receptor? So in other words, what system in the cell can read this zip code and interpret it correctly to ship the proteins where it has to go? Then, more molecularly speaking, what's the structure of the translocon or the translocation channel and how proteins are moved? So especially for proteins that need to move across membrane. So you remember the first lecture with Carlos, right? There are biochemical constraints here. Proteins in the cytosol, they would be hydrophilic, right? Soluble. But they have to go across across membranes, which have this hydrophobic core, right, of fatty acids, right? You're supposed to know that already. Yes. Well, or you're still learning that. Fair enough. Right, so there's a conundrum here, how a soluble protein can cross a membrane. So this is why it's a big deal. And often, this is a so-called endergonic process, something that requires energy, right? And so it's always a question as well, where does that energy come from? So signal sequences, I'm not in love with these kind of lists, so we'll go quickly through that. So these signal peptide or targeting sequences, they're usually short sequences of amino acids that are always found in the protein themselves, and importantly, they work just like a zip code, and we're just going to see a few examples. So today we're going to focus on translocation at the ER. So I'm showing you this micrograph to kind of anchor all these concepts in reality. This is obviously electron microscopy. And what I'd like you to be able to do eventually is, given an image like that, without the annotation, to recognize what you're looking at. And so not just name the parts. So for example, the parts would be, well, you clearly have a membrane, electron dense, right? Absorbs electron in electron microscopy, especially the hydrophobic core. On the outside, and it forms a compartment. So that's the lumen, and that's the membrane forming the endoplasmic reticulum. And you would even characterize it as being the rough endoplasmic reticulum because you see these structures here, and those are the ribosomes. Okay, and some of them are hanging loose in this medium here. That's the cytosol, right? So fairly straightforward. And so these are the parts. Now the meaning of this image comes from the fact that you see all kinds of ribosomes very tightly associated with the ER membrane. So in a snapshot, you have a view of the process we're going to talk about today, which is this co-translational translocation, whereby ribosomes associate with the ER membrane as they translate proteins and help push proteins through the membrane into the ER. Okay, so those images have a meaning. Okay, so in cartoon forms, this is what I'm talking about. The idea is that if you have an mRNA that encodes a membrane protein or a secreted protein, right? Again, we're going to pause for a slight second here just to make you think about it. I told you the zip code is in the protein. So there's no system to read the mRNA and know from the mRNA where the protein is going to go because the information is in the protein. So somehow translation has to start, right? Translation always starts from the N-terminus. That's a universal truth of translation in molecular biology. So for most of these proteins that are being secreted, the signal is actually at the N-terminus, right? In other words, and this is kind of depicted in this cartoon, once the protein begins to be synthesized, the first amino acid stretch that pops out is the signal. So now the machinery can read that and act upon it. And one of the first actions we're going to see, what do you think it is? It's getting more complicated. Well, the first is to pause translation, stop translation, and bring the complex of the RNA and the ribosome to the ER membrane. Okay? So if you think about this, you have to think about some soluble receptor that will have to recognize these nascent transcripts here and somehow help pause translation and bring the whole complex of the mRNA, the nascent protein, and the ribosomes to the ER membrane. Okay? So this has been studied, and I'm not gonna spend too much time on this, but it's important for you guys to have some sense of the kinds of experiments that were done and can be used to study these sort of processes. So this was studied some time ago using biochemical work, and here is a depiction of the system. So the idea is that these proteins are being synthesized, and if you give the cell these radio-labeled amino acids, so this S35 cysteine, for example, these will be incorporated into newly synthesized proteins. Okay, so you can, in this way, somehow study the dynamics of the system. Now, the ER is very complex, but the trick was to break it down, into smaller parts called so-called microsomes. And the important point is that these microsomes are functional. They have all the necessary components to allow for this co-translational translocation to happen. So they can now be studied in vitro, and they recapitulate what happens in vivo. Okay? Does that make sense? And some of the simple experiments that were performed are the following, and you can hopefully see how this works. So imagine you collect a suspension of such microsomes, and they contain some of the newly synthesized proteins, which have now incorporated these S35 cysteines, and you split your fraction in two. One of them, as indicated here, you treat it with a detergent. So remember from Carlos's lecture, what do detergents do? They solubilize lipids. Okay, so now you no longer have two compartments inside the microsomes and outside the microsomes. It's all solubilized. Okay? And so in one fraction you don't treat, and then what you do, you add protease, a protein that will not be specific, but will basically chew all the proteins it can access. Okay? So the difference is that fraction here, the protease will digest the newly synthesized proteins, and here, so what you would do after that is you run a Western blot, for example, or some kind of protein gel, and you try to identify your proteins of interest. So after this treatment, the expectation is if proteins have been translocated into the microsomes, they should be protected when you don't add the detergent, but they should be degraded when you add the detergent. Okay? So fairly simple. All right, so using these kinds of methods and others, I guess, this is how the system was somehow dissected some time ago. So the idea is that indeed the signal peptide for ER translocation is always located at the N-terminus. I explained to you why. It's a short amino acid. It's like 16 to 30 amino acids. Usually, there's a stretch of hydrophobic amino acids, and that's going to be important because, again, if you think about it, hydrophobic amino acids popping out of the ribosome in a very aqueous cytosol, there's a thermodynamic problem, right? You need something also hydrophobic to quench that, ideally. Okay? I'm going to get back to that. And eventually, and we'll see, I'm going to insist upon that, also because it's a hydrophobic sequence, once the protein enters the ER here, that signal peptide is cleaved off. Okay, that's a recurring theme as well. And in this so-called hydropathy profile, you can always detect them. So what is this? Let me explain what you're looking at here. So this would be the primary sequence of a protein, here the human growth hormone receptor, right? So it's more or less big. And in the y-axis, this is hydropathy, or the opposite of hydrophobia. Oh, sorry, of hydrophilia. Okay? Sorry, this is hydrophoby. So if it's positive, that means those amino acids tend to be hydrophobic. All right? So what this is showing us is that there's this stretch here of hydrophobic residues right after the methionine. Right? So that's telltale for the signal peptide. Okay? And then we're going to talk about that at the end of the lecture. There's another stretch of hydrophobic residues here. So what do you think that can be? I know, I'm talking fast. Am I talking too fast? No? It's the transmembrane domain. Right? You remember, again, I want you to make connections. That's why I bring up these other lectures, because you saw transmembrane proteins, there's a strong thermodynamic constraint to have the residues, at least some of them, be hydrophobic. Okay, so, and you can tell on a hydropathy profile. Let me just stop this. Okay. So, what's the receptor? I know you're all dying to know now, right? You were like, tell us about the receptor, right? So here it is. So, what you want in this receptor, you want something that can do many different things, okay? Because it has to recognize the signal peptide. So the receptor, let me give out the name, it's the so-called signal recognition particle, very creative. Because it can recognize the signal and it's a particle. You can come up with your own favorite name if you want to, just not in the exam. But when you talk about yourself, you can call it a Gizmo 1, and then you say, hey, remember Gizmo 1. So anyway, so the whole idea, right, is that this particle is basically diffusing in the cytosol. And sometimes, often, it encounters a signal peptide coming out of a ribosome. And then it latches onto it. Okay? So that's what's depicted here. And sure enough, there is a domain here. This is a complex structure. There are multiple proteins, and there's a scaffolding RNA that helps keep it together. And the idea is that this domain here, P54, helps recognize the signal peptide. But like we said, this is not the only thing that this particle has to do. It's one of the things. The other two things, at least, are stop translation, right? It's not enough to just hang on to the signal peptide. You got to stop translation. So there are domains here to interact, interact with the ribosomes, right? So this is really, at the molecular level, a multifunctional particle or complex, if you will. And finally, we talked about how one of the jobs of this SRP is to bring the whole translational complex to the ER membrane, right? So there is this other domain here required for translocation, and the idea is that at the ER membrane, there's a cognate receptor, right? Which is not recognizing the signal peptide directly. It's recognizing SRP, right? So SRP is the mediator between these translational complexes and the receptor here. All right, and so again, it's always good to think about structure-function relationships. So I told you the signal peptide has a stretch of hydrophobic residues. That might cause a thermodynamic problem if it comes out in the cytosol. So indeed, the signal peptide, the signal recognition particle, and in particular, the domain that recognizes the signal peptide, like in this case, this structure here, which is from a bacterium, but that's analogous to this P54 subunit, has a hydrophobic binding group, right? So if you had to design it, that's what you would do. You would say, well, I have an hydrophobic signal peptide coming out. I want to quench that to stabilize the system. I need what? A hydrophobic pocket to attach to. Okay? Okay, and indeed, so a lot of the knowledge and that is in the textbooks now has been established using this microsome paradigm again, right? So for instance, when this was used, this was determined that the signal peptide is cleaved off once the protein gets in, and actually very quickly. So as the protein starts to be translocated, once the signal peptide has already made it, it can be cleaved off, right? The system, and here what's depicted here is that, see, this signal peptide is already being removed and destroyed, but the protein is still being synthesized and still being translocated. Yes?
So does P54 back to the other, so does P54 bind to the sequence on the mRNA, or is it the protein?
No, on the protein. It binds.
Because the slide before it showed like
Really?
binding to RNA.
This?
On the right. Or on the right.
Oh yeah. Hey. Okay. Sorry. So this is not the messenger RNA. This RNA, good question, is a scaffolding RNA. So what do I mean by that? It's part of the structure of SRP, right? And so it basically holds all the different domains together. But it's not the mRNA that's being translated and that's producing the protein. Okay? All right. So let's move on. Okay. So using this paradigm, so what this slide is basically illustrating is the following. Is if somehow you manage to synthesize the protein in vitro, and there are ways to do that, right? You assemble ribosomes with mRNAs and so on. So you can presumably synthesize one of those secreted proteins in vitro. You could argue, well, actually, I don't know, because maybe the signal peptide here is going to be destabilizing the whole structure. It's possible, but you can still get a protein. And if you try to do that, and only then add it to microsomes, those proteins never made it inside the microsome. And you can test that how? Using this detergent and protease assay. Okay, so this was very well established. And indeed, so it has to be co-translational, right? Back to this concept. The translocation has to happen at the same time as translation. So let's go back to the process itself. I mean, I really try to unpack this for you. I think in the textbook it's like one figure, but there's a lot to unpack in this process. And so here is one of my favorite elegant aspect of the system. And by the way, when I say this is one of my favorite things, you know what that means. I ain't kidding. Okay, so But I mean, you love it too because it's so elegant, right? So here are some of the problems that the system has to solve, right? We talked about energy. We talked about pushing the proteins through this hydrophobic membrane. But the first thing is now the SRP is docked onto the receptor, so it's now associated to the ER membrane. But the protein is not being translocated yet, right? So this cartoon does a good job at showing you that the signal peptide is still engaged with the SRP. Don't get fooled by this. There's no cleavage. This is an error in the drawing. The translocon, we'll talk about that, is closed. So whatever channel, the gate through which the protein has to go is not open at this stage. Okay? But you now have assembled the SRP particle carrying all the complex with its receptor. And what I like about this is that each moiety, so the SRP subunit and, the SRP receptor and the SRP itself, they each carry half a GTPase domain. So what does that mean, GTPase? It's something that's going to hydrolyze GTP. What do you get if you hydrolyze GTP? Energy. That's right. So you get a functional GTPase domain only upon association of the two moieties. So now you have something that won't waste energy, that can harness the energy of ATP only upon association of the SRP with the receptor. Okay? So what do you think is that energy going to be useful for? Opening. That's right. Opening the sorry, I'm going to calm down. Opening the translocon, right? You need to open the gate to allow the protein to go through. And that's exactly what happens. So upon hydrolysis of GTP, now the protein itself is handed over from the SRP receptor to the translocon, which has opened, right? And the signal peptide goes first. This cartoon is trying to represent the fact that because it's a hydrophobic sequence, it tries not to go, but eventually it will. And the other aspect is that the SRP itself is recycled, right? So upon hydrolysis of GTP, the complex and release of the nascent protein to the translocon, the complex deassociate, and the beauty of this is that now SRP can be recycled, right? Go back and snatch another nascent protein, okay? All right, so what happens next? So let's just recap for a second here. The signal sequence is this N-terminal hydrophobic ER signal peptide. The receptor is the SRP and the receptor. The energy, I talked about GTP hydrolysis, but in this case, the translation itself also helps power the translocation. Basically, imagine if you have a ribosome that's docked on a translocon and it's pumping out the protein, that protein gets almost passively pushed through the translocon. Of course, okay, so to some extent, the energy required to translocate the protein also comes from the translation itself. So now let's look a bit more at this translocon itself. It has, yet again, a few very elegant features at the molecular level. So very briefly, this was used using a complex system whereby you can engineer proteins and you engineer artificial mRNA and tRNAs, and then you use complex cross-linking and biochemistry to try to dissect which part of the translocon interact with translocating protein. I'm going fast through that because, how do I put this? This is not one of my favorite aspects. Okay? Now, back to the concepts, right? To how things work in reality. So in these crystal structures, or these 3D models of the structure, now that's the translocon. And there are at least two features, well, let's make it three. Three features that are interesting to think about and recognize. The first one is, if you look at this one, does it look familiar to you? Think membrane proteins. First lecture or second lecture. Alpha helices getting together, forming a structure within the membrane. That's classic, right? So the translocon has that. A bunch of alpha helices, which on the outside can interact with the hydrophobic core, and on the inside can interact to variable extent with the protein being translocated. Okay, but it has a couple more features. One of them, right, the cartoon showed translocon closed, translocon open. This, in a structure world, boils down to this plug here. So this is a small bit of amino acid that can, swing in and close it or swing out. Right? So this is a conformational change. And what do you think can power this conformational change in moving from closed to open? Is the GTP hydrolysis, exactly. Okay, so now you're starting to connect the dots, right? And there is one last feature which is really neat, and I'm going to highlight now and get back to in due time. So this is a side view. Imagine you're now in the membrane runs in this way. Imagine now you're looking from the top. So you can see this the pore ring here. So this is where the proteins go through. So these residues in green, they are the ones to interact with the proteins being translocated. But there is a lateral exit, and we'll see this is very important, right? So there's a way, and I'm going to just prime you to think about that. We talked about proteins going through. Well, that's great for a protein that's going to be soluble in the ER lumen. But there are a lot of proteins that have to stay at the membrane. And so for those, there's this lateral exit door. Okay, so the translocon has kind of a hole and proteins can translocate through, but if they stop, it can also open on the side and let the protein go through in the membrane. And we'll see how that matters. Okay, so now the translocon is open, the protein is engaged upon GTP hydrolysis, and translation helps push it through. Of course, as this goes, now you need this intra-ER peptidase. So this is also a transmembrane protein, but it has this soluble domain on the ER side, and that helps recognize and cleave off the signal peptide. Okay? And finally, the protein continues to push through, and it needs to be folded. So this is not depicted on this very slide, but there are proteins on the ER lumen side called chaperones, which help take care of the newly synthesized proteins and fold them properly. Okay, and those are essential for the proper folding and function of these proteins. All right, so to recap, I've told you quite a few things already. Right, and that's just the first part of the first lecture about the secretory pathway. There's a lot to unpack here, right? So we saw how the nascent protein very quickly exposes this signal peptide, leading to recognition by the signal particle, which pauses translation and brings the whole complex to the ER membrane, interacts with the receptor, and upon GTP hydrolysis helps open the translocon, transfer the protein to the translocon, and deassemble the complex releasing SRP. I forgot to highlight that, but some of you may have already anticipated that the release of SRP is also important for what? Translation to resume. That's right, because its presence blocked translation initially. So the releasing it, deassembling the SRP ribosome complex is very important for that. So and it doesn't happen until this protein is engaged in the translocon, right? So if you had to design it, you would not do a better job than natural selection did. It's very unequivocal. Okay, so let's move on. So in the rest of the lecture, there are a few, variations to this theme that I'm gonna basically illustrate. So one of them is somehow contradictory to what I said in the first place, which is, whereas most of these proteins have to be translocated co-translationally, right? So this is something you might want to practice doing, in the shower in the morning, like co-translation or translocation. It's not easy to pronounce. Imagine me, I have to teach that. So I have to be very eloquent about it. Otherwise, you guys fall asleep. That's the problem. So sometimes some other proteins can nevertheless be translocated non-co-translationally. Okay? And so in this case, you have the proteins is fully formed. There's a problem about keeping it unfolded, and we're going to see examples of that in other lectures. And somehow it can still interact with the translocon. This is essentially things you found in yeast, for example. But you have to now power the translocation, and that involves these chaperones in the ER lumen, which use ATP to hang on to the protein and basically pull it in. Right? So I'm just gonna, right? So imagine the chaperone hangs on to the bit of protein coming in, hydrolyzes ATP, pull, the next one, the next one, the next one. Okay? That's one alternative way. It's not the main way. Okay, so to summarize, we saw the signal peptide, the SRP and the receptor, or in this case, the translocon itself. In the main case, the translocon is the transporter. It's slightly different in this later case I just briefly mentioned. And in this later case, ATP hydrolysis powers translocation, whereas in the most common case, it's both translation and GTP hydrolysis. So quite frankly, the very first part, what's in red here is what I really want you to focus on, right? That's the core of how things work in eukaryotic cells. Okay, so now let's shift topic a little bit. It's kind of the second half of the lecture, and we're going to now talk about transmembrane proteins. So I want you to think about it from a kind of structure-function relationship, and I made this very sophisticated animation for that. So you have the nucleus, right, with two membranes, the nuclear envelope. It's connected to the ER, but I depicted it this way, then the Golgi, then the secretory vesicles, and the lysosome. So imagine, and at the plasma membrane, of course, you have a lot of proteins. What kinds of proteins can you think about in terms of plasma membrane proteins? Blood type protein, for example, receptors, right? Those are very important, and they are countless. I mean, not countless, but there are thousands and thousands, okay? And there is something very important about the function, which is, and that's the orientation, right? If you, let's say, a MHC complex, well, you need the information to be exposed to the outside, right? So you want the protein, oops. I gave away my magnificent animation. It has to be with this domain exoplasmic and this other domain cytoplasmic. That's what's called the topology. And you could have the opposite. Okay, but the opposite would just not work. So you have to get it right. Now, the problem is you put the protein at the membrane here, at the ER. I just showed you this. Okay? And then the protein is anchored at the membrane, and there's no swapping the topology. So the topology, which matters for the function, whether at the plasma membrane, the lysosome, the Golgi, and so on, is established upon translocation at the ER. Okay? And so hence the beautiful animation here. It doesn't change. Okay? Because once the way things are trafficked, and we're going to see that, is proteins are packaged into vesicles. But these proteins, once they associate with the membrane, they stay associated with the membrane. They can't swap topology. Okay? So let's look into the topologies. As a matter of fact, you have all kinds of possibilities. And now we are talking almost exclusively about, yes, you have a question? Don't be shy. I don't bite. I mean, I haven't bitten anybody yet. No? Okay. So these topologies, right? So the simplest one we can think about is this one. Is because we saw how every protein, when it gets translocated, it's the N-terminus first, right? I didn't insist on this, but it's a consequence of everything I've told you so far. N-terminus comes first. So to have this topology here, where you have only one transmembrane domain, and you have the C-terminus hanging out in the cytosol and the N-terminus making it into the ER lumen, all you need to do is start translocation, cleave off the signal peptide, those have a signal peptide, and then you just stop translocation once you have the transmembrane domain. Okay? Then the opposite topology, where now you have the N-terminus towards the cytosol and the C-terminus in the ER lumen. We're going to have to think about that and see how this can be achieved. And then finally, this one, well, it's very similar to the first one, except most of the protein is in the cytosol. Now, the type 4 is what confuses your predecessors every year. So, no doubt it's going to confuse some of you, and we're going to try to really unpack that and make it clear how this works. Like, how can you get multiple transmembrane domains in the same, connected in the same protein, if you have kind of a unidirectional translocation system? We're going to see how this works. And I'm going to end with these so-called GPI anchored proteins. And here are examples of each one of those. Needless to say, I do not need you to remember all these examples of proteins in this way, right? Just understand how this is done. Okay, so let's go through them kind of one by one. Right, so this now you should be familiar with this cartoon. The protein is being translocated and co-translationally. So as the nascent parts go through the translocon, if this is now a type 1 transmembrane protein, single pass. So single pass means there's one transmembrane domain. What happens is, once the transmembrane domain goes through the translocon, and you can tell it's here, it's a stretch of hydrophobic residues, right? That's what you expect for a transmembrane domain, especially a single one, right? If it's a alpha helix, it has to be hydrophobic residues all around, okay? Now, what happens is this so-called stop transfer anchor, or STA. It's just useful for you to remember the different terms we're going to use here. The name tells you what it is. Stop transfer. That means it's ongoing transfer and it stops, right? And it stops in part because it's a stretch of hydrophobic residues. They resist going into the hydrophilic phase of the ER lumen. Okay? But what do? Are they getting stuck in the translocon? Do you want that? No. But they don't want to go into ER lumen. So they use the lateral door, the so-called lateral exit to the lipid bilayer, right? And now they leave the translocon. Okay, so you have a deassociation between the translocon and the ribosome that's continuing to synthesize this protein. And sure enough, the translocon closes, and now the rest of the protein is synthesized outside. So the C-terminus remains in the cytosol, and you're done. Now you have a protein that's a single-pass transmembrane protein, has its N-terminus in the ER lumen, and the C-terminus in the cytosol. So that protein, if it's now trafficked all the way to the plasma membrane, which part will be facing the outside of the cell? Exactly. What cytosol, I mean, one way to remember it is what cytosol here is going to be cytosol all along. Right? Because if you think about the ER lumen as this compartment, and then vesicles are made and trafficked, then eventually when it fuses to the plasma membrane, it goes like this. Right? So what's exoplasmic so the term exoplasmic refers to what's in the ER lumen, because this is the compartment, this is the side that's going to be outside later on. Okay? And what's cytosolic here remains cytosolic all along. Okay, so here these proteins will expose the N-terminus to the outside if they are trafficked all the way. Okay? Okay, so now things get a little bit more complicated because it's different from what I've told you so far. It uses the same machinery, but the topology is not the same. So how does that work? So imagine now you have this single-pass transmembrane protein, but it wants to have the opposite topology. The N-terminus is on the cytosolic side, and the C-terminus is on the ER lumen or exoplasmic side. Right? So these cartoons show it very well. One very important point here. These guys do not have a signal peptide. So in the cytosol, the ribosome will synthesize this protein. And then at some point, this transmembrane sequence will show up. That's a hydrophobic stretch of amino acids. And it will recruit SRP. So the same SRP machinery will bring this protein to the translocon. And the translocation will happen. But now the topology, the direction in which the translocation will happen is not so obvious, because there's already a stretch of N-terminus protein here. So which way is it going to be oriented? This is determined by the presence of a stretch of polar and actually positively charged amino acid. So if you go back to your biochemistry, at cellular pH 7.2-ish, some amino acids are charged, right? Like they have NH2 is not going to be NH2, it's going to be NH3+. Okay? So if you have a stretch of such amino acids before the now signal anchor, now this is called a signal anchor again, the answer is in the name because it acts like a signal, except it's never going to be cleaved. It's going to become a transmembrane domain. Right? So the topology is given by the presence of this signal anchor and the presence of this positively charged amino acid. And the logic is the charged amino acid always remain on the cytosolic side. Okay? So the machinery knows to insert this protein in the translocon in this way, right? And then as translation continues, this is the C-terminus that's now being pushed to the translocon, and sure enough, the signal anchor is not being pushed through because it's a stretch of hydrophobic residues, it's a transmembrane domain. So what happens to it? The lateral exit. Right? So the translocon opens up to let it go into the membrane, and that's how, so these cartoons do a good job at showing that, right? You have now the transmembrane domain is into the membrane, but the C-terminus of the protein is still engaged through the translocon and being pushed through. And you end up with a single-pass transmembrane protein with the opposite topology. N-terminus is now on the cytosol, the C-terminus in the ER lumen. Right? So, yes?
How does the translation work without the special enzyme, the S?
So it's not represented in this cartoon, but SRP also recognizes the signal anchor.
Oh, okay.
Yeah. So it's not drawn here,
Yeah.
but it's the same SRP that does the job of recognizing the presence of this signal anchor. So that's why it's called a signal anchor. Right? The main difference with the signal peptide, the N-terminus signal, is that it's not translocated through and it's not cleaved. It remains in the protein and it becomes the transmembrane domain. Okay? All right. So, yeah, so I'm just going to go super quickly through this because so this one is so-called type 3. So you see the topology is the same as the type 1. So you think, why bother? Well, because the mechanism is not the same. Yes.
Is this also hydrolyzed by GTP?
Yes. You mean the previous one, the type 2?
This one,
Yeah, this one as well. So it's not depicted here, but the idea is that the same complex of SRP receptor, GTP, co-translational translocation, the same mechanism happens here. The main difference with what I spent half an hour talking about is the topology, right? So the type 1 is kind of the default topology. Here you have to encode alternative topologies using this positively charged amino acid. Okay? So this one, I call it, it's not alternative. You see it's the same as the type 1, where you have the N-terminus on the ER lumen, but the difference is that there's no signal peptide. Okay? Somehow these proteins don't have a signal peptide, but they have a transmembrane domain, which can serve as a signal like we saw. So this is also a so-called signal anchor. But now you want the topology with the N-terminus inside. So the simple solution is to have the positive residues on the other side, right? Because again, those positive residues will always remain on the cytosolic side. And you can think about it as they are positively charged. The translocon likes slightly hydrophobic sequences. So there's almost an electrostatic repulsion. I mean, it's a simplification, but it's one way to remember it. Okay? Positive stays out. And so you have such proteins exist, again, and you find a stretch of amino acid of hydrophobic residues, but that's not the beginning of the protein, and the beginning of the protein, and this stretch also is never removed, right? Again, because it becomes a transmembrane domain, right? So now we can move, now we've understood a few basic rules to place and orient transmembrane domains for proteins. We've only looked at single pass. So how do multipass do it? Well, okay, so that's just the summary, blah, blah, blah. The type 1 signal peptide gets cleaved off, stop transfer anchor, lateral transfer. This one, signal anchor, recognized by SRP, bringing it, and the orientation is determined by the position of the positively charged amino acid, which always remain on the cytosolic side. Okay? I'm just eager to move to the multipass. The multipass transmembrane proteins. So they are super important, right? I mean, translocon is one of them. Many receptors are multipass transmembrane proteins. Cells can't do without them. But they have to place them at the membrane. Right, and actually, so this one is a good example. GLUT1, you know what this is? Did Carlos tell you about the GLUT? So those are, sorry? It's not glutamine, good try. It's glucose transporter. I don't know why my accent became so thick all of a sudden, but glucose transporter, right? For glycolysis, it's essential. The cell needs to import glucose, and glucose is a charged molecule, so it doesn't diffuse passively through the membrane. You need a transporter, and GLUT1 is one of such transporters and is a so-called type 4 transmembrane protein because it has multiple alpha helices. And again, to help you guys connect the dots between the lectures, when you want to create a transporter for a hydrophilic molecule, sometimes even a charged molecule like an ion, you need, within the membrane, which is otherwise hydrophobic, you need to create an environment that's going to be hydrophilic. And one way to do it is to have multiple alpha helices getting together, and those alpha helices have a side that's hydrophobic, that interacts with the hydrophobic core of the membrane, and another side that's hydrophilic and that can help create a pore if you use multiple helices. Okay? Again, you would not have designed it better. So, but now the question that occupies us today is how are those positioned at the membrane? So, in cartoon form, so in hydropathy profiles, you see these stretches of hydrophobic residues along the primary sequence. In cartoon form, this one, for example, is going to start with the N-terminus in the ER lumen, and then a transmembrane domain, and a transmembrane domain, and a transmembrane domain, and so on. And there are two kinds. And that depends essentially on where the N-terminus is. Because once you've determined where the N-terminus is, whether in the cytosol or in the ER, then it imposes a strong constraint on the topology of the subsequent domains, right? I mean, if you think about it, it's like you weave it through the membrane. So you can't have two successive transmembrane domains in the same orientation. The topologies have to alternate necessarily. So to put it simply, this uses the exact same logic I described for the single-pass transmembrane proteins, except it does it over and over again. So in my experience, the best way to really understand this and see how this works is to think about the dynamic process. So imagine, because all of these are translocated co-translationally. So it always starts with the N-terminus and then progresses towards the C-terminus, right? So in this case, for example, so there's no signal peptide. There can be a signal peptide, but it's not needed. The first signal is a signal anchor. So SRP recruits it, okay? Brings it to the translocon. The positive charges are on this side, so you know that side is going to stay in the cytosol. Yes?
Are the transmembrane domains the ones that are actually in the membrane, or like the ones that are just like the ones that are like looping the two that are in?
Yeah, usually transmembrane refers to the stretches that go through the membrane, so this would be the stuff that's in the membrane, right? So that's depicted in red on these cartoons, right? And in this zigzag thing, okay? But basically, if you think about it dynamically, this one gets put at the membrane, and this stays in the cytosol. So once this is transferred laterally, this stretch here is now translocating, right? So it's actively translocating. And then the next one comes, so it's necessarily a stop transfer. So now the protein leaves the translocon. This stretch is synthesized in the cytosol. And now the the signal anchor shows up. So another SRP particle gets it, brings it to a new translocon, and translocation starts over again and lets it go once it's through. And now this stuff goes through. So it winds up in the lumen and so on. Right? So that's why I'm saying, if you think about it dynamically, like how is it weaved through, you actually understand the topology. And there's no way you can alter this. If once you've determined the beginning, you have to have everything fall in place. Okay? And normally the position of the positively charged residues tell you always this stretch of protein here will be in the cytosol. Okay? And if you apply the same logic here, but with the opposite topology, you'll see it works just fine. Okay? So it's a good idea. I mean, I will post actually homeworks. Probably not today because I'm traveling to give a talk somewhere else. But so you can practice this, and you will practice this because it's one of my favorite things. Okay, so just to finish a few additional examples. So this one, for example, is one way to get. Okay, so how do you get it? How do you? That's not a pun. How do you place a protein to the membrane? Yes?
Is it possible for a terminus and C-terminus to end up on the same side?
Yes? How would you do that?
Oh, I don't know.
You add another transmembrane domain. Yeah.
Is there like another side? Like,
So you would basically have, because many of those have 7 transmembrane domains. So it's a good question, actually. If you want to do that, you basically need even number of transmembrane domains. Right? So if you have only two, and let's say the N-terminus is an ER, you have the first one, the next stretch is in the cytosol, and then the second one, and the C-terminus is going to be in the ER as well. Okay? So if you have so many proteins have an odd number of transmembrane domains, especially 7, the classic 7 transmembrane domain G protein coupled receptors is just like that. But in principle, and I just can't think of an example right now, but you could have an even number of transmembrane domains, and then you would have that. Does that make sense? Yeah. Okay, just to finish quickly, this one, it's kind of an aside, is what do you do if your transmembrane domain is actually the most C-terminal stretch? In other words, the machinery has no way of knowing that this is going to be a transmembrane protein until the protein is fully synthesized, because it's the C-terminus. Okay, so that's this example. So you have a machinery to recognize this, and this uses now directly ATP to power the system, and it uses a special kinds of receptor because there's no real need to translocate, so to speak, right? You just kind of have to plug that protein into the membrane. And some of those are actually achieved through lipid modifications, and that's what I'm going to end with. It's also, I have many favorites. This one is also one of my favorite things. The GPI anchors, just because it's so important in biology, and it's really cool. So let's pause a second to look in awe at this structure. It's beautiful. Why is it beautiful? Because, so how would you call such a molecule? It has 2 long chains of fatty acids, and then this connection through a phosphate, and with now these sugars, and inositol, glucosamine, and mannose, and so on. It's, well, actually, it's a good call. It looks like a phospholipid. A more generic term would be an amphipathic molecule. Okay? Because it has a hydrophobic side, the fatty acid chain, and a hydrophilic side. So where would you expect this protein, it's not a protein actually, to be anchored at the membrane with this part, the sugars sticking out, the polar domain sticking out either on the, on a hydrophilic compartment. Okay? Now, this is about the side of a hemimembrane, right? The 2 halves of a membrane. So it's only going to be anchored on one side of the membrane. So now, you've probably seen this with Carlos as well. How do you think this stuff will move in a membrane? Like easily or kind of getting stuck? Easily, right? High mobility. So now you have something that can be anchored in the membrane, but very loosely. So it can be very mobile in the membrane. So wouldn't it be cool if we could now functionalize this GPI anchor by attaching proteins to it? And that's exactly what the cell is doing, right? And so those kinds of GPI anchored transmembrane proteins are very important. So this cartoon shows you how this happens. Is that, so the GPI anchor is always in the ER, well, starts in the ER membrane and is facing the the ER lumen side, okay? And you have proteins which are themselves already anchored. So those proteins, right, you have to think of them as they've been translocated co-translationally before, but they have a transmembrane domain, except because the way proteins are, this transmembrane domain spans the entire width of the membrane. So it's probably going to be less mobile, right? So somehow there's this GPI transamidase, and what it does is cleave off the protein and transfer the functional N-terminal domain onto this GPI anchor. So now you have a so-called GPI anchored protein, and you can surmise that it will be much more mobile in the membrane. And most of these proteins, as a matter of fact, they get trafficked through the secretory pathway, and they wind up at the plasma membrane exposed to the outside. Okay, and some of those actually carry blood groups and so on. And before I let you go, 5 minutes early, I'm sure you won't be mad at me for that. I just want to give you a primer for future lectures. These sugars, they tend to form these complex polymers here. I mean, this one is not that complex, but and this always happens on the exoplasmic side in the ER and in the Golgi. And as we'll see during one lecture, there are a lot of so-called glucidic modifications of proteins, always for secreted proteins, always on the exoplasmic side, and therefore always on the domains that become exposed to the outside. Okay? And I'm going to stop here.